Ladies and gentlemen, it is my great honor to speak here. I appreciate again all the member of this KSSCR and all participants. As introduced, I'm in the moment the president of JSRM. This is a very large and unique society for develop the regenerative therapy. And so that I would like to, of course, introduce the JSRM. But before introduction of JSRM, at the beginning, I would like to introduce my work, and then I will introduce JSRM. As you can see here, that we have developed the cell seed technology so that we can realize such beating heart seed with Professor Okano in Tokyo Women's Medical College School. He is already retired, but he is my previous president of JSRM, and also our collaboration started 2000, so that these 17 years we have developed, and finally we could make such heart seed for the commercial available level. As you know, that about 20 years ago, the regenerative therapy proposed, so that such kind of the bone marrow cell injection directly into the heart seems to be the new paradigm for heart failure treatment. And until now, so many kinds of cell sources, such as bone marrow cells and cellular cell or myoblasts or several kinds, are applied. Probably until now, more than 300 protocols in the clinical trial are applied. But until now, no project are approved as commercially available level. Of course, to find the cell source, it is very important. But to how to deliver the cell into the organs, it is also very important. Until now, we developed such kinds of the cell sheet technology or cardiac graft. This technology started 2003, and in the moment we are applying for the clinical level using the autologous skeletal muscle cells. Once these cells are applied for the heart failure patient, so-called very famous trial, the MAGIC trial, but it was failed in Europe, and so that in that time the myoblast was not suitable for the regeneration of the heart. But we changed the technology, how to transplant or how to introduce to the heart from the direct injection to cell seed. We could get this great success. For example, you can have a look of this is a preclinical trial using the canine heart failure model. This model is, of course, dilated myelocardiopathy. But after the implantation of autologous myoblast cell seed, we can realize the treatment or decrease the size of the heart and improvement of the thickness of the wall, as you can see here. And finally, we could get, of course, improvement of cardiac function. And in case of MAGIC trial, as I told that myoblast is harmful to occur lethal arrhythmia. But using this technology, no lethal arrhythmia have been detected. Then we moved to the clinical trial in 2007. And for example, first case, Under left ventricular assist device, he is waiting for the donor heart, but we try to implant this skeletal muscle myoblastic seed on his heart under assist device. And finally, he could get the weaning from assist device, and already 10 years has passed, but he is still very active in the moment. Of course, he was a DCM patient, but it was very effective. And this patient is an ICM patient. She is very young, but she is also waiting for the left ventricular assist device and transplantation. After this treatment, he seemed to be very, very healthy. And after this clinical trial, we have done more than 50 cases for DCM or ICM, and we could get, for example, improvement of survival. For example, for 5 years survival, as compared to the predictable survival curve or heart transplantation curve, we could get the improvement using this autologous myoblastic seed. During this activity, you know that Japan changed the regulation, so-called expedited approval system under PMD Act. In case of regenerative therapy, it takes much more longer time as compared to the chemical product, so that Japanese government fix for the new pathway for the regenerative therapy using the conditional time-limited marketing authorization. After a short time of the clinical trial to prove the safety, we could get the conditional time-limited marketing authorization. And on market, of course, it takes 2-3 years. But on market, we have to prove the evidence of the efficacy. This is very good because so that the scientific level is just same as a normal clinical trial. But after short-term trial and after authorization, on market, we can get the approval, the final approval. This is the earlier access, and also that for the developer, patient, and academia, it is win and win. It is very nice. And also, under this new regulation, Terumo, you know, the Japanese medical company, started the clinical trial of our project using the autologous skeletal myoblastic seed in 7 cases in 3 centers. And it takes, of course, if we count since 2000, it takes a long time, but finally we could get the approval from the government, and also Terumo has started the heart sheet, and it is we can apply commercially available level. But of course, still there is a limitation because skeletal myoblasts is not so powerful because this is cytokine therapy. Skeletal myoblasts can produce HGF, VGF, FGF, SDF1, or several kinds of cytokine. This is, of course, has the potential to regenerate, but for patient side, patients should have the still viability of the heart. It is very important because if the patient has no lower level of the viability, it does not work well. So that there is a responder and a non-responder. Therefore, this is of course the first step for our trial, but the second step, we are now planning to start the iPS cell seed. Probably almost all cases seem to become the responder for iPS cell seed because the iPS can deliver the cardiac cells and also cytokines. As you can see here, that already we can make the very powerful cardiac cell sheet using the iPS cell-derived cardiac cell sheet. And these success, we can already have the proof of concept. In 2012 already, using the human iPS cell sheet, we can treat the big myocardial infarction, as you can see here, and also improvement of the cardiac function. And the most important thing is we can detect the maturation of the cardiac cells. When we implant the cell sheet, it shows still the immature level because we derived from the iPS cells and we get cardiac cells, but still it is immature. But after implantation, after 2 months, we could find the maturation. So that this is much more, we can have the expectation of the more powerful recovery of cardiac function, as you can see here. So that summarize of the mechanism by different cell source using the same technology, cell sheet. But if we change the cell source from the skeletal myoblast, in case of skeletal myoblast, of course, they can deliver HGF-based something kind of cytokine. But if we change the cell source from to iPS cell-derived cardiac cells, they are still mature so that they can produce much more cytokines, more suitable for the heart regeneration. And also the iPS cell cardiac myocyte replenishment. This is also very important. So that finally we could get the much more higher efficacy of the improvement of cardiac function or remodeling. Already we are now preparing, for example, we need high volume cultures and for human, and also the GMP level culture are needed. So that in the moment in Osaka University, we are now preparing. And finally, we could get the evidence to implant the cells safely and efficacy. And just now, we submit this protocol to the committee. Of course, since 2000, we started as a heart sheet, but since 2008, we started the iPS cell sheet. And during that time, in 2012, Professor Yamanaka got the Nobel Prize, and then government give a big project to Kyoto University to make a very safe iPS cells for clinical application. And we received already such clinical, such safe iPS cells for clinical application. And in the moment, we are discussing PMDA, and now we are now planning the first in human in 1-2 years. So that this is media's reports, so many kinds of the media reports about our submission of our protocol to the first-class certified special committee for regenerative medicine. So that I hopefully half year or 1 year, I will start the clinical trials using the of course, allograft, but iPS cell-derived cardiac cell sheet. These are my activity, and just now, I would like to start the introduction about JSRM. As you know, that the JSRM is a very, very unique and world's largest regenerative medicine community. Its activity is very high, and I would like to introduce our activity. In 2001, we started our society, and the mission or vision is to deliver safe and effective regenerative medicine rapidly to the people. This is our mission. So that the JSRM has been actively promoting research on regenerative medicine and the development of therapeutic methods using regenerative medicine based on fulfilling the society vision. Our most unique stance is welcoming every stakeholder concerning in regenerative medicine and the technologies around. So that, as you know, that we have developing year and year, and in the moment nearly 6,000 members already we have, and 10% growth every year since 2011 we can have. And once in these members, of course, more than half is academia, but nearly 1/3 is the member of the companies. This is also very unique because industrial side also take part to this society. So that our goal is to achieve to get the success for the regenerative therapy as a universal therapy. And also, so that our unique action is launching policy proposals, including opinion of regenerative medicine industry. This is very, very unique because in 2012 in Yokohama, we have the Yokohama Declaration. In that time, I was a Congress president, and we present this declaration because we hope to change the regulation. Until that time, regulation was only for chemicals, drugs, and also medical device, not for regenerative therapy. But regenerative therapy is now developing and developing, so that we should change. This is the most bottleneck to get regenerative substance, so that we hope to change the regulation we access to the government and the government people with brainstorming and discussion. And finally, our proposal understood by government, and the new national framework for regenerative medicine in Japan was started in 2014. At first, Regenerative Medicine Promoting Act was established in 2013. And then under this act, two acts newly come. One is the Act on the Safety of Regenerative Medicine for Clinical Study or the treatment outside of the insurance. So that it is also very, very important because there is no regulation for such cases to promote and ensure regenerative medicine. And the second, Pharmaceutical and Medical Device Act was changed, including the including regenerative medicine. This is, of course, a regulation gets approval from the government, and most important is company. So that the both act is very, very powerful and innovative in the moment in the world for develop the regenerative therapy. And the first act for safety, we have such three types of the committees. One is a high risk, so-called class 1, for example, including this category, iPS cells, ES cells, or the allograft are discussed. And this is in this case, in the special certified special committee are discussed, and after permission, then again everything discussed in the government. But in class 2, we don't need a discussion with government or approval with government. And also class 3, this is very low risk autologous cells using, for example, PRP or something like for the cosmetic treatment or something like also are included. But the important thing is keep the safety, so that in this meaning, this act works very well. On the other hand, the pharmaceutical affair law, in the affair law, until now, there are, as I told, that only for the drugs and medical device, but additional division is a regenerative medical products. And also, as I told, that in the time, conditional term limited approval system are introduced. As I explained this law. And also, as you know, that we every year, every month, we propose to government to make this this law, so that in the world are surprised. And for example, the Stem Cell Summit give us the Stem Cell Action Award in 2014, or for example, the Nature Medicine have interest in their paper, or several kinds of the companies, or the USA Ambassador Kennedy invited me to ask what happened. So that in the moment, in the moment, it is just a turning point to make the new law, to make the new cell source or new technology, so that until now almost all cell source are autologous, but in the moment we should change from autologous to allogenic to get the great success or new breakthrough to the new area for regenerative therapy. So that now is a very important time. And also we have the such articles, manuscript, as you know, that the regenerative therapy, so that please submit your paper also to this regenerative therapy. Now, finally, I would like to introduce new strategic action with government. So as I told that to make the regulation, we work together with government. In the moment, the government are very interested to improve the regenerative therapy, especially the industrial side. So that for this action, we are now building out the regenerative medicine national consortium to cover whole Japan. Our society just started to make every resource open and accelerate clinical research. So that at the beginning, for our role, to promote or accelerate the clinical research, this is important. But of course, not only for clinical research, but also for the industrial side, it is very important so that smoothly it should be continued. And the National Consortium Act is included support for clinical researches, development of human resource, development of clinical research data system, and the coordination of academia, industry, translation, and also interactive communication with patient citizens. This is very, very important. So that our new mission is a nationwide platform to make regenerative medicine safe, effective, and concrete as soon as practical. So that I don't say so much because I don't have time, but the main role is to support the clinical researchers by several kinds of the main institute, and to make a kind of JSRM has become a kind of a national-wide COE, Center of Excellence. And also, development of human resource, we have make so much effort. Since 2014, authorization system for certified physicians and cell processing operators, we have already started. And also, the most important thing is to make a national regenerative medicine database. And we get from PMDA, we got computerized system validation system included database. And this is this is most important for the good post-marketing surveillance, PMS, so that the company, any company, when they hope to get the proof after permission or PMS, every company have to use this national database. And so that we should make such database as a very scientific and precious data. And also we have already started academia industry translation, and also interactive communication with patients or citizens. This is also very, very important for the ESCAR level. So for such activity, now we have just started for these 3 years. And I hope, again, I would like to say thank you very much for invitation. And so that the KSCR is a very, very active society, as I know, in Korea, so that if possible, we should have the good partnership with KSCR and the JSRM. Thank you for your attention.